It's our pleasure to welcome back for today's webinar Dr. Eric Brodsky, Associate Director, Labeling Policy Team, Office of New Drug Policy and the Office of New Drugs, CEDAR. Please join me in welcoming our presenter for today's webinar, Dr. Brodsky.
Thank you for the kind introduction. I would also like to welcome you to day 2 for a discussion about the dosage administration section of labeling draft guidance. As I stated yesterday, this guidance was published in January of this year, and there were two main reasons why we revised it from the last version, which was published in 2010. Number one, to account for the increased complexity of dosage and administration related information in labeling over the last decade, and also given that we have about 20 labeling guidances that provided recommendations about how to include information about dosage and or administration in labeling. And we wanted to incorporate those recommendations in one place as well, which is the DNA section of labeling guidance. This is my disclaimer. So this is what we discussed yesterday on day 1 of the presentations. We talked about the general principles for developing the dosage and administration section of labeling, recommended organization and format with respect to tables, bulleted lists about what should appear first generally in the section and what potentially could be appeared later, critical dosage and administration related information that appears prior to the fundamental dosage administration. So critical dosage and administration information would be, for example, recommendations to dilute a product when the undiluted product can cause toxicity, or for example, to avoid a certain route of administration if that route of administration is associated with clinically significant adverse reactions. We also talked about the fundamental dosage information, the recommended dosage, the starting dose, how to titrate the dose, maximum recommended dosage, dosage in specific populations, as well as the recommended dosage for fixed combination drug products and co-packaged products. Again, these were the topics for yesterday's presentation. So today, we're going to talk about several topics. We're going to talk about administration instructions that are included near the recommended dosage, such as the route of administration, or potentially administration with food or food substances for an orally administered product, recommended monitoring for effectiveness, such as therapeutic drug monitoring, other therapy that is used prior to the subject drug, and medication that is used concomitantly with the subject drug, dosage modifications due to adverse reactions, and dosage modifications due to drug interactions. Additionally, recommendations on how to discontinue a drug when a drug is associated with a withdrawal syndrome on abrupt discontinuation, like a benzodiazepine or an SSRI or a beta blocker. Administration and preparation instructions for certain products. And finally, storage instructions for the prepared product, like the diluted solution or the reconstituted solution. So as I stated yesterday, we basically sponsored a study of 70 physicians. About 35 of them were generalists and 35 were specialists. And these were 45-minute interviews, and we asked them how they obtain drug information, what do they prefer with respect to the prescribing information, how do they understand specific information in the prescribing information. And one of the questions that we asked them, these 70 physicians, is that which sections of the prescribing information were most useful to their clinical practice. Now, we show them all up to 18 sections, the 17 sections and the box warning sections, and we asked them this question. Physicians were allowed to pick more than one section. And the highest rating sections appear as shown. And the dosage administration section of labeling, at least for this study of 70 physicians, that was the most useful for these physicians. So obviously, the dosage administration section is an important section for healthcare practitioners. As I stated yesterday, this is a picture of the first page of the draft guidance. It was published in January. I looked at all the comments so far that was submitted. Thank you for the comments. Please, if you have additional comments, you're welcome to submit them to the docket. And we will consider all comments prior to finalization of this guidance. As I stated yesterday, the purpose of this guidance is to assist applicants in developing the information in the DNA section of labeling and to ensure that this section contains the dosage and administration related information needed for the safe and effective use of the drug. As I stated yesterday, we have nearly 20 examples of information in the DNA section of labeling. Previously, when we wrote guidances 10 years ago, we didn't provide that many examples, but we find that the examples may be helpful. They're helpful for review teams within FDA to review and develop labeling, as well as helpful for the applicants. So administration instructions that are included close to the recommended dosage. So if there are specific instructions, this is for an orally administered product, or orally like a tablet, a capsule, oral solution. If there's specific instructions about how to use such products relative to the ingestion of food or food substances, it should be in the DNA section of labeling. And generally, we recommend it next to the recommended dosage. So this is an example. So here we include the recommended dosage, and in this example, we say take with or without food. Now, this is actually, in terms of this recommended language, some of this is from the recommendations from those 70 physicians. One of the big concepts that we heard from them is that their patients frequently ask them for an orally administered product, can they take it with or without food? So even if there's no recommendation to avoid food, or to take it on an empty stomach, or to take it with a low-fat diet, they recommend including this specific information. If you could take the drug with or without food to include that specific statement in labeling. And that's in a prominent location to help physicians answer those questions for their patients. This is another example about including administration instructions with respect to food. Again, you include the recommended dosage, and then you include the recommendation here is to take it on an empty stomach at least 2 hours prior to or 2 hours after food. Now, the data that supports this recommendation is in another section. It's in the clinical pharmacology section, particularly pharmacokinetic subsection 12.3, under the absorption heading under the food effect subheading. This is another example. You include the recommended dosage, and then you include specific instructions about taking it with a low-fat meal. And then the data, again, is in the pharmacokinetics subsection 12.3. Now, sometimes, uncommonly though, food effect recommendations are not based upon pharmacokinetic or pharmacodynamic data. And in that case, you would cross-reference another section of labeling, typically, for example, Section 5 and potentially Section 6, with respect to the rationale. So, for example, if you have a product where it's shown that you take the drug with food, that reduces the incidence of nausea or vomiting compared to taking the drug without food. The information about the why, the details of that would be in other sections of labeling, like Sections 5 and 6, but just the high-level recommendation to take it with food would be in Section 2. Now, with respect to intravenous administration, again, this is included along with the recommended dosage. According to the regulations, we must include in the DNA section specific directions for the administration of the dosage form. So in this example, we would include the rate of administration and the recommended infusion duration. This is a rather lengthy infusion. It's over 5 hours. It provides the starting initial intravenous infusion rate over an hour, and then subsequent infusion rates. A titration scheme. Intravenous bolus versus intravenous push. Generally, intravenous push is administered over seconds. Intravenous bolus is over minutes. So if you are, the recommendation is to administer the drug by intravenous bolus, we recommend providing more specificity. In this case, you administer over 2 minutes because that can range. So provide those specificity to those folks administering the product. Now moving on to intramuscular or subcutaneous administration. In these routes of administration, we want to provide the recommended injection sites and or locations, and then the rotation schedule. So this specific example provides 4 different specific locations that one would take it, and then the rotation recommendations. So this is another example of intramuscular or subcutaneous administration. In this situation, you need more than 1 injection to achieve the full dose. So here, the recommended dosage is 300 milligrams given subcutaneously every 3 months. However, the only strength is 150 milligrams. So you need to give 2 150 milligram subcutaneous injections. There's no 300 milligram strength. So you have to identify that in the DNA section of labeling. Additionally, we provide information about how to do that. You give it here at least 2 inches from the site of the first subcutaneous injection. So additional information about the location of both of these injections. And finally, for intramuscular or subcutaneous administration, sometimes the injection depth or the duration of the injection is important, specifically if the duration is lengthy. Now, a classic example of this is a monoclonal antibody hyaluronidase product. Some of these products are administered over several minutes. So if that's the case, the recommendation is to include, at least for the first part, the injection depth, if that is really important for administration, or the recommended duration of the injection. That's the monoclonal hyaluronidase example. And here, if you could see the example, we administer the drug subcutaneously over approximately 8 minutes, which is a rather lengthy subcutaneous injection. So these are administration instructions for modified release dosage forms such as extended release tablets, delayed release tablets, or extended release capsules, and delayed release capsules. So if there's data that demonstrate that there's a risk associated with manipulating the drug, one would include the following or similar statement. And the concern here is that if you overcome the extended release or delayed release properties of the drug, you may get a total influx of the drug, essentially much greater exposure right after administering the drug, and that could lead to a clinically significant adverse reaction or risk. So in this section, we include do not split, crush, or chew the extended release tablets, and the rationale is included in another section of labeling, the clinical pharmacology section, specifically the pharmacokinetics subsection. Now again, this is for modified release dosage forms, so if we don't have specific data, but we still are concerned that we may modify, that modification may alter the drug's safety or effectiveness, we can include this general statement. Again, this is for modified release dosage forms. Swallow tablets whole, avoid splitting, crushing, or chewing extended release tablets because doing so may compromise the extended release characteristics, effectiveness, or safety of drug X. So chewable tablets, dosage form, these are specific administration instructions. So according to USP, chewable tablets must always be chewed. It may not be swallowed, must not be swallowed. So if a product may be chewed or swallowed, then the dosage form is tablets, not chewable tablets. So if you have a chewable tablet dosage form, USP recommends including specific information in labeling regarding the chewable tablets characteristics. And the DNA section of labeling guidance recommends including specific language such as chew or crush drug X completely before swallowing. Do not swallow the chewable tablets whole. And the rationale behind this is included in the pharmacokinetics subsection 12.3. Now let's move on to a different dosage form, tablets for oral suspension and tablets for oral solution. These are a little less common. And these are tablets that there's a little bit more flexibility. So they are intended to be dispersed in liquid prior to administration, but they could also be chewed or swallowed whole. And so USP says that we should provide the methods of administration in labeling. The DNA section of labeling guidance provides a little more specificity. That should be in the DNA section of labeling guidance, again, for these specific dosage forms. And here we state these tablets for oral solution is the dosage form, may be dispersed in water or fruit juice, that's the liquids, swallowed whole, or chewed. So we're providing more specificity to the healthcare practitioner about how these products can be administered. Now, system dosage forms. So a system dosage form is a delivery system that controls the rate of the drug. Typically, commonly, it's a transdermal system. But we have other types of systems, vaginal systems, uterine systems, ocular systems, periodontal systems, oral mucosal systems. So for these examples in the DNA section, we should provide the rate of release. So here in the example, we say transdermal system, and in the parenthetical, we say delivers 5 milligrams of drugazide over 24 hours. We should provide the total duration of the drug release. That's in red. As well, we should provide specific information about how to apply the product, where to apply the product, potentially how to rotate administration of the product, how to remove the product, and potentially how to reapply the product. That's all in this example. So moving on to a slightly different topic, recommended monitoring for effectiveness. This is commonly done with therapeutic drug monitoring. So this is to maintain effectiveness, but as well reducing the risk of adverse reactions. And examples here are cyclosporin, digoxin, gentamicin, lithium, phenytoin, tacrolimus, valproic acid, vancomycin. Those are drugs in which we use therapeutic drug monitoring. So in this situation, you want to provide the concentration window. So if you go above that concentration, you may get toxicity. If you go below that, you lose efficacy. We also want to provide recommended frequency that you monitor, and then dose modifications. So if it goes too high, what do we do? If it goes too low, what do we do? So this is an example of a drug used in the setting. It's an immunosuppressive in patients with a recent kidney transplant surgery. This is basically, we're measuring concentration, and the dose, the concentration windows differ according to the time out from surgery. So after surgery, you have higher concentrations that are recommended, And towards later after surgery, you have lower concentrations that are recommended. And our jobs are to try to ensure that the concentration are within these ranges during those times. And we titrate the dosage to maintain these concentration ranges. Here's another example of therapeutic drug monitoring. In this example, we provide specific information about when to assess, when to monitor the concentration. This is 7 to 10 hours after the morning dose and 4 weeks after initiation, and then periodically thereafter. And in this situation, we're targeting 300 to 1,080 nanograms per deciliter. If it's within that range, the concentration, we continue the dosage. If it's lower than that range, lower than the lowest number, lower than 300, we want to increase the daily dosage. If it's greater than the highest number, 1080, we decrease the dosage. So we try to maintain that 300 to 1080 concentration window. So other therapy used prior to the subject drug, as well as concomitant therapy. So if there are important dosage and administration related information about other drugs before initiating the subject drug, that should be included in the DMA section. So for example, if you have a product associated with hypersensitivity reactions, and we have data that suggests that including pre-medication lowers the risk of hypersensitivity reactions, the specific pre-medication regimen should be included in section 2. And again, that's generally, because this is critical information, generally it's included prior to the fundamental dosage and administration recommendations. And you also do it before administrating the subject drug. When we provide this information, this is specificity, the specific names of the drugs, their dosages, as well as the timing of these drugs prior to administration. Notice for the first drug, we administer at least 1 hour prior to the drug X infusion. The second drug, we administer at least 30 minutes prior to drug X infusion, so they're slightly different. The details about cytokine release syndrome here and then hypersensitivity reactions are included in section 5. However, the pre-medication is included in section 2. Now, required concomitant therapy. So if we have a drug that is used in an indication only in conjunction with another therapy, for example, another drug, we must include that information in the indications of usage section. We must say that the subject drug is used in combination with these other drugs. That's a requirement. So what do we do in this situation in section 2? In section 2, we also should name those drugs. So here, the recommended dosage of drug X in combination with drug A and drug B is 10 milligrams given intravenously once weekly. Now, we did have a really good question in the docket about how much specificity should we provide about the concomitant drugs. So generally, what we do is reference the prescribing information of the other drugs. So we would say here potentially refer to the prescribing information for drug A and drug B for the dosage recommendations. Why do we do that? Because sometimes these dosage regimens are complex. There's dosage modifications due to drug interactions or adverse reactions, where there's a different dosage recommendation in patients with renal impairment, and it's hard to include all of that in the subject drugs labeling. So we refer out to the other labeling. However, sometimes this is potentially a new disease, a new indication that we're treating, and drug A, drug B's labeling doesn't have dosage recommendations for that. Or these drugs were dosed differently in this regimen. And it's important to tell people what that regimen is. So in that case, instead of referring out to the other prescribing information for drug A and drug B, for those dosage recommendations, you would include the specific regimens for drug A and drug B. Now, with respect to preparation and administration instructions, we would only include preparation and administration instructions for the subject drug, not drug A and drug B. That information would be provided generally in the other drugs labeling, and we would reference the prescribing information of drug A and drug B to find that complex preparation instructions. So dosage modifications. So as I stated yesterday in yesterday's webinar, we use different terminology whether we're talking about a recommended dosage versus a dosage modification. So for specific populations, we use the term recommended dosage. And the example I gave yesterday is patients with renal impairment. If you have chronic renal impairment, the next day you're not going to have normal renal function. So you're really not adjusting the dose in that one patient. It's a different recommended dosage. So again, when we're talking about the recommended dose, we talk about the recommended dosage in patients with renal impairment, the recommended dosage in patients with hepatic impairment, the recommended dosage in patients that are a certain CYP4 metabolizer. We use the term recommended dosage. In contrast, when we're talking about adverse reactions or drug interactions, those are dosage modifications. So you take the recommended dosage, and then you have an adverse reaction, then you modify that dosage. That's called dosage modification. Similarly, you're taking a drug, and then someone takes another concomitant drug, you're modifying that subject drug dosage. As I stated in yesterday's presentation, typically we include dosage modifications for adverse reactions and for drug interactions later in the section. Typically, we include the critical information about dosage and administration. Then we include the fundamental dosage information, including the recommended dosage. And then later, typically, we include dosage modifications for adverse reactions and drug interactions. And I should say dosage modifications could be a dosage reduction, a dosage interruption, or a permanent discontinuation. Now, dosage modifications intended to reduce the risk of adverse reactions. So this is an example. This is most commonly for oncology products, although other labeling include dosage modifications due to adverse reactions. So this is an example for oncology drug. As I stated earlier, we want to provide a recommendation to introduce the table. So we say, see table 1 for drug X dosage modifications for adverse reactions. In the table, it's complex information. A table can be helpful. We include the adverse reaction as well as the severity. There are different dosage modifications according to the severity. Here we're using the CTCAC, which is the common terminology criteria for adverse events. And so someone could go to that and understand what a grade 2 is, a grade 3, grade 4, with respect to that specific adverse reaction. So we know what specific adverse reaction is and what's the severity. And the third column provides the dosage modification. So that could be a dosage reduction. It could be holding the drug and then potentially resuming it when you have a complete or partial resolution, or a permanent discontinuation. Now, I should say that details about these specific adverse reactions, pneumonitis, colitis, should be in section 5, for example, and section 6, but it should not be, the details of these clinically significant adverse reactions should not be stated in section 2. Section 2 could cross-reference to section 5, for example, for those details. So dosage modifications, again, to reduce the risk of adverse reactions. In this case, we have hypersensitivity reactions in section 5. So again, the details of these adverse reactions are in section 5. And then in section 2, we just provide what to do. So the why, again, is in section 5, and what to do is in section 2. And in this case, in section 2, we have a slightly different recommendation. We have a different recommendation for severe hypersensitivity reactions to modify the dosage. You stop the drug. If you have a mild to moderate hypersensitivity reaction, you basically reduce the dosage, which is slowing the infusion rate or reduce the dosage to this effect. So with respect to, again, about location of information about tests, procedures, and evaluations that are needed during treatment or before treatment. So if there's tests, procedures, or evaluations that are needed during treatment, but it's got nothing to do with modifying the dosage or changing the administration, that information is included in other sections of labeling, such as the warnings and precautions section, rather than the dosage and administration section. However, if there's a required or necessary information about test procedures or evaluations that are needed before drug initiation, like, for example, if you want to verify a pregnancy status, doing pregnancy testing in a drug associated with embryo-fetal toxicity, and you want to do this testing prior to administration, information about the testing should be in Section 2 and generally in the beginning of the section. Additionally, if you have tests and procedures that could result in modification of dosage and or administration, that should be included in Section 2, in addition to how to modify the dosage, whether you decrease the dosage, you temporarily stop, or you permanently discontinue. That's how you think about organization of how this information appears in labeling. Moving on to dosage modifications for drug interactions. So dosage modifications for drug interactions, again, this would be a dosage reduction, alteration of the timing of a dosage relative to dosing another drug, and if we have sufficient information to support a specific recommendation to modify the dosage or administration of the subject drug to reduce the risk of a drug interaction, this should be, it actually must be included in Section 2. Now, we would only include the recommendation to modify the dosage or administration of the subject drug. The other drug, if we're modifying that dosage, that information would be either included in the drug interaction section of the subject drug's labeling, or we refer out to that other labeling in the drug interaction section. So the DNA section of the subject drug concentrates on the subject drug. So you're only going to provide the modifications of the subject drug due to the drug interaction. So similarly, as I stated in yesterday's presentation, we want to provide the complete picture of dosage recommendations, this specifically dosage modifications for drug interactions. So if you have a dosage modification in a specific drug interacting class, so let's say you have CYP3A inhibitors, and there's a specific dosage recommendation modification for moderate use with moderate CYP3A inhibitors, you should provide generally the recommendations for the use of the subject drug for the remaining subgroups. So you want to provide the dosage modifications, if any, or the use of that drug with strong CYP3A inhibitors, as well as weak CYP3 inhibitors. This is for completeness. So this may be a case that we may modify the dosage. We may reduce the dosage when taking the subject drug with moderate CYP3A inhibitors. However, with strong CYP3A inhibitors, the recommendation is to not use the drug, not recommended with concomitant use of strong 3A inhibitors, and that should be included in this section. We should also include information about weak CYP3A inhibitors. The only caveat is that it's not necessary to include a statement about that there's no dosage modification needed for a class where it rarely requires dose modifications, such as CYP3 weak inhibitors. So if there's a weak CYP3 inhibitor, you don't need to include that modification. But generally, the principle is if there's a modification for one subgroup, moderate CYP3 inhibitors, one should provide recommendations about strong and weak CYP3 inhibitors in the DNA section for a complete picture. So this slide is about how potentially to develop the subsection title if there are dosage modifications due to drug interactions. So in the first example, if there's a single drug or a single class or a single food, one can consider including that drug, class, or food within the subsection title. So for example, if the single class is CYP3A inhibitors, one would use a title such as dosage modifications for CYP3A inhibitors. However, if you had two or more drugs or two or more drug classes or foods, you can consider having a broader title, such as dosage modifications for drug interactions, and have headings under that subsection, and each of the headings would include the specific drugs or classes or foods in which there are dosage modifications. So certain things are required in Section 7, the drug interaction section, if there are clinically significant drug interactions. One is a description of the clinically significant drug interaction. If the mechanism of the drug interaction is known, that is stated. So if you look at the last row, that includes that information. The mechanism, drug oxide, is a CYP3A substrate. That's the mechanism. The description of the clinically significant drug interaction, strong or moderate CYP3A inhibitors increase drugoside X exposure, which may increase the risk of drug X adverse reactions. It also includes the clinical implications or the clinical effects. Additionally, one includes the prevention and or management strategies. Now, if there is a change in dose, if there's as I stated earlier, if one of the subgroups, let's say moderate CYP3A inhibitors, there is a change in the dose, then one refers to the DNA section for the specificity about how to modify the dose. There's a general statement that's included in Section 7, and that is referral to Section 2 for the details. Now let's go to Section 2. As I stated, that you want to complete the entire picture. The dosage reduction is with moderate CYP3A inhibitors, and you see the dose reduction to 100 milligrams once daily when used concomitantly with moderate CYP3A inhibitors. And we want to complete the entire picture, so we also include information about the use of the drug with strong 3A inhibitors, and it's avoid concomitant use. And as I stated earlier, one wants the complete picture, and the one exception is if there's a subgroup, let's say weak CYP3 inhibitors, in which dosage modification is rare, one would not need to include that subgroup. However, generally, one wants to include a complete picture about usage of the drug if there's dosage modifications in one of the subgroups. So we're switching gears right now. We're going to talk about specifically for drugs associated with a withdrawal syndrome, a discontinuation syndrome, on abrupt discontinuation, or potentially a very fast taper, and what information should be included in Section 2 with respect to these situations. So for this example, let's start with Section 9, drug abuse and dependence, and specifically subsection 9.3, dependence. So this subsection, if there's a withdrawal syndrome upon abrupt discontinuation or abrupt dosage reduction, one describes the discontinuation syndrome here. And the recommendation here in the drug abuse and dependence section of labeling guidance is to refer to the DNA section for the specific tapering recommendation to reduce the risk of a withdrawal syndrome upon abrupt discontinuation. So this is what Section 2 would look like, discontinuation of drug X, and when you're discontinuing the drug X, how you decrease the dose. until you discontinue the dose. But again, the details of the withdrawal syndrome is included in Section 9. The specific tapering schedule is in Section 2. And these subsections cross-reference one another. Moving on to preparation instructions. Preparation instructions are required as appropriate in Section 2. So when, as appropriate, one should identify compatible diluents, for example, to dilute a product or for reconstitution. And if you were reconstituting, you provide the volume of diluent needed for reconstitution. And I'm showing the example below. So identifying the compatible diluents, that's sterile water for injection, as well as the volume for diluent, that's in green. So when one identifies the diluents, the recommendation is to use the established name and strength of the diluent. And I give you a few examples here. So one would use 0.9% sodium chloride injection instead of normal saline or saline. And I listed two other examples of where one would include the established name of the product, if there's a USP monograph, rather than other terminology in Section 2 when describing the diluent. So preparation instructions. This is a table that provides additional information with respect to if a drug is reconstituted only, diluted only, and reconstituted and diluted prior to administration. It's important to provide the strength of the final dosage form in terms of milligram of active ingredient per milliliter. So if you're only going to reconstitute a product, for example, if it comes in a lyophilized powder and you reconstitute it into a solution, one should identify the volume of the reconstituted solution that is withdrawn and then subsequently administered. Similarly, if you're only diluting a product, one should identify the amount, the volume specifically of the diluted solution to be withdrawn and administered. However, if you have a for ejection, let's say, dosage form, and that is going to be first reconstituted and subsequently diluted prior to administration, one does two things. First, identify the volume of the reconstituted solution to be withdrawn that will be subsequently diluted, and then the volume of the diluted solution that is withdrawn and administered. So again, reconstitution first, then dilution, and then administration. So this is for a product that is stored in a refrigerator or freezer. This could be the supplied product. It could be the diluted product. It could be the reconstituted product. And the key message here is that you're allowing the product to warm to room temperature. You're not warming the product. You're allowing the product to warm to room temperature prior to administration. And the recommendation, the guidance, is to provide the time, how much time one needs prior to administration. And in this case, in this example, it's 30 to 40 minutes to sit at room temperature prior to administration. This example also recommends not using an external heat source to heat the product because heat can damage the product potentially. So if you have a prepared product as stated in the refrigerator or freezer, a diluted product or reconstituted product, it's important to provide the length of time that you could keep the diluted or reconstituted product at room temperature prior to use, as well as the appropriate discard instructions. And this example talks about that. So after removal of the drug X reconstituted solution from the refrigerator, use the reconstituted solution within 2 hours or discard. These are specific cases of preparation and administration for certain products. So if there's specific containers such as glass or plastic or specific devices, certain tubing or needles or syringes, and that those are needed for preparation or administration, one should provide the specificity about the specific containers or devices. However, if containers or devices will not be approved under the NDA or BLA, the recommendation is to describe the device or container in general terms rather than being very specific. This will allow the healthcare practitioner to use many different types of containers or devices as appropriate. If there is some information about incompatibility with respect to the use of the drug with a specific container or device, that information should be provided. Essentially, state what containers or devices are incompatible with the drug. So for some oral dosage forms, some of these products may be indicated in neonates, infants, or other younger pediatric patients. And in those cases, those younger pediatric patients can't swallow. And if you qualify a liquid or soft food to be mixed, prepared, and then administered, these patients can receive the drug. So what do I mean by liquid? That could be apple juice or milk or infant formula. Soft food vehicles could be applesauce, bananas, rice pudding, strawberry jam. And generally, in the example I show here, one should include the specific qualified liquid or soft food. In this case, it's applesauce, as well as the amount of liquid or soft food. And here it's a teaspoon or 5 milliliters to a tablespoon, which is 15 milliliters, as well as how to manipulate the product, carefully open the capsule and sprinkle the entire contents of the capsule onto applesauce, stir the mixture for 10 seconds, as well as providing information about storage or lack of storage. Here, the recommendation is not to store the product. Additionally, if certain liquids or foods are not qualified and the recommendation is to avoid such foods or liquids, one should provide that information here, specifically also the rationale, such as if the liquid or soft food dissolves protected coating and destroys potentially the drug substance. So sometimes we have products that can be administered by enteral tubes. If we have adequate data to support the use of oral dosage forms, such as a capsule, granules, oral suspensions, via enteral tubes, such as an NG tube, nasogastric tube, one should include information in the DNA section about how to prepare it and administer the product via the enteral tube. And in such cases, one would provide the characteristics of the enteral tube, the preparation instructions of the tube, as well as the drug, how to administer the product, and after administration, how to maintain the enteral tube. So this is a good example of how to include preparation and administration instructions of an oral dosage form via an enteral tube if there's sufficient data to support this. So I highlighted a few elements in different colors. In blue, we identify the type of tube. It's a nasogastric tube, French size 8 or larger. In green, we identify drug preparation instructions. Red has administration instructions. And the purple has some administration instructions as well as instructions on maintaining the tube after administration. So storage instructions of the prepared product, for example, diluted product or reconstituted product. One question that frequently comes up when reviewing labeling is how to include storage instructions within the labeling. So for example, storage instructions of the supplied product versus storage instructions of the prepared product. So our regulations, specifically the House Supplied Storage and Handling Section regulations, Section 16 regulations state that one must actually, as appropriate, include storage conditions of the supplied drug. So that's the unopened package. So Section 16, if we didn't touch it, that's the storage instructions. However, the DNA section, the regulations state that one must contain specific direction on preparation of the dosage form. And so here, the DNA section includes information about the diluted product storage and the reconstituted product storage. So again, supplied product, unopened package, section 16, manipulated product, prepared product, diluted, reconstituted, section 2. So storage, so what do we provide with respect to storage instructions for the reconstituted or diluted product in section 2? We should provide the storage conditions needed to maintain the stability and administration conditions of those products, and then also the length of time in which the reconstituted or diluted product can be used under the storage conditions, and then an appropriate discard statement. And on the next slide, I'll show you an example of this. So this is the example. This is storage instructions for a specific reconstituted product. In purple, the specific instructions about how to store the product at room temperature and for how long, and how long to refrigerate the product, as well as discard instructions that are in blue. Challenge questions. So challenge question number 1, what information about adverse reactions should be included in the dosage administration section? And I'll give you a few seconds to read this question as well as look at the answers. So dosage reductions intended to reduce the risk of adverse reactions, that information should be included in section 2. Now let's go through the other choices. A, detailed description of the clinically significant adverse reaction or adverse reactions must be in section 5, the warnings and precautions section. It could also be in other sections of labeling, like section 6, adverse reaction section. But again, for the DNA section, we want a concise section, and detailed descriptions should be in other sections of labeling, like section 6 and section 5. Now, dosage reduction intended to reduce the risk of adverse reaction is one way to manage an adverse reaction, but we don't include all the steps that take to prevent, mitigate, monitor for, or manage the adverse reaction in section 2. Again, we want that very concise so we don't distract or compete with the required and recommended information in section 2. D, well, we should include dosage reduction intended to reduce the risk of adverse reactions. Adverse reaction data from the clinical trials are generally in section 6, the adverse reaction section. They're not included in section 2. Challenge question number 2. What information about drug interactions must or should be included in the DNA section? So answer B is correct. Dosage modifications due to drug interactions. In this case, it's a specific class, CYP3A4 inhibitors. Now let's go through the other choices. Descriptions of the clinically significant drug interaction must be in section 7, the drug interaction section. That is not included in section 2. Clinical implications of this clinically significant drug interaction, generally we include that in section 7, drug interaction section, not section 2. And B, D, and B, E are incorrect choices. So challenge question 3, which answer is incorrect? So let's go through the correct answers. A, B, and D are correct. We include dilution and reconstitution instructions in the DNA section. That's A and B. Answer D, we do include storage instructions about the supplied product in section 16, how supplied storage and handling. Answer C is not correct. We include dilution instructions in the DNA section, not in the how supplied and storage and handling section. So closing thoughts. So when you're submitting labeling, whether this is an NME labeling, new biological product labeling, or you're modifying an existing labeling under a labeling supplement or efficacy supplement, please ensure the DNA section contains all the DNA information needed for the safe and effective use of the drug. I also say that consider if information in the DNA section would be more appropriate from other sections of labeling so we don't distract or compete with the required and recommended information in the DNA section. And finally, please, if you have any comments or additional comments, you're welcome to submit them to the docket about the DNA section of labeling guidance, and we would like to consider that prior to finalizing the guidance.
Thank you for another great presentation, Dr. Brodsky. We'll now transition into our Q&A session. If you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. Looks like we have some questions coming in right now, and here's the first question for Dr. Brodsky. If submitting a labeling supplement that wouldn't typically have changes to dosage and administration, is it acceptable to update the labeling to improve format and organization of the sections like dosage and administration at the same time per recommendations here and in the draft guidance?
Good question. Yes. And not only is it acceptable, we would encourage you to update the labeling to be consistent with the recommendations and the guidance. This obviously, as stated earlier, the DNA section labeling is an important section. It's useful for healthcare practitioners. It can potentially reduce medication errors. So labeling is an iterative process. There's always something potentially one could update. So if you see something that could be made more clear, yes, please do that. Thank you.
Thank you for responding to that question. We do have more questions already. And here's the next question. Are there any considerations if pre-medication is only required in a small subset of patients or certain populations?
Good question. So in that case, one would construct the DNA section of labeling by stating that pre-medication is recommended in subpopulation A and provide the specific pre-medication for that subpopulation, and also state for subpopulations B and C, pre-medication is not needed. Thank you.
Thank you for responding to that question. We do have more questions available. Here's the next question. How important is it to state that a drug is to be taken orally when it should be taken orally? I've seen drugs that have been have to be taken orally just described as to be taken once daily.
Good question. So it's very important. So first of all, administration instructions are required in the DNA section of labeling. So from a regulatory standpoint, route of administration is part of administration. But but additionally, we've had medication errors about using drugs in inappropriate routes of administration or inadvertently using it for other routes of administration. So, for example, previously we used to call the vaginal tablets. Now these products are called vaginal inserts. So instead of taking these tablets, these products, I should say, these inserts vaginally, people were taking them orally. Sometimes we've had tablets that are inserted in an inhaler, and you're supposed to take them for oral inhalation. People have been taking them orally. Sometimes we have topical products that people take orally. So maybe for that, one may think, well, for this drug right here, well, maybe it's not an issue. But thinking about the number of prescriptions people take, thinking about actually specific from the healthcare practitioners, how many prescriptions um, they do per day. It's important to specify the route administration, even though it is obvious. It did for tablets, for capsules, extended release capsules, extended release tablets. Yes, we do recommend using the term orally for tablets. Thank you.
Thank you for responding to that question. Here's the next question. For a drug with multiple capsule or tablet strengths, where the recommended dose can be achieved in more than one way, such as 100 milligram tablet or two 50 milligram tablets, is it recommended to list all options, or is a more general statement preferred, such as dose may be achieved with any combination of tablets?
Good question. So if in this example, if the recommended dosage is 100 milligrams once daily, then one does not need to include information about the strengths in the DNA section. We have a section 3, dosage forms and strengths, and one's required to include the strengths. One is also required to include the strengths in the house supplied storage and handling section of labeling. So, and potentially it could be confusing and distracting. So, healthcare practitioners are sophisticated. They could figure out if the recommended dosage is one is 100 milligrams once daily. They could see it's supplied in 100 milligram tablets or 50 milligram tablets. They could decide how to prescribe that drug. So, one does not in that scenario need to include the strength of the tablets in the DNA section. Thank you.
Thank you for responding to that question. We got another question on a dosage adjustment, recommended dosage, and here's the question. If there is no dosage adjustment for mild impairment, but dosage must be modified for the moderate impairment, can we state that no dosage adjustment is required for mild renal impairment, or do we have to repeat the dosage again in this section, even when no modifications are needed? Also remind us, please, if the preferred phrase is dosage adjustment or recommended dosage in patients with renal impairment.
So I'll take the second question first. So, as discussed yesterday, the preferred phrase is the recommended dosage in patients with renal impairment. We don't recommend using dosage adjustment or dosage modification in patients with renal impairment because generally, the patients with chronic renal impairment, they've had that for a long time. So, in that particular patient, you're not adjusting the dosage. They have a they have they their creatinine clearance may be 56, 60, and that's been going on for a long time. You're not adjusting the dosage in that patient. That's a specific recommended dosage. So that's part two of the question. Part one, with the case that um, you're providing a different recommended dosage in those with moderate renal impairment compared to those with normal renal function, but the dosage in mild renal impairment is the same as those with normal renal function. So there are a couple options here. In the example that I provided yesterday, is that I gave the specific dosage recommendation in those with mild renal impairment. However, one could also state that the recommended dosage in patients with mild renal impairment is the same as those with normal renal function, and then cross reference to the other part of the DNA section of labeling that includes the recommended dosage in patients with normal renal function. So there are two ways to approach that question. Thank you.
Thank you for responding to that question. We got another question related to renal impairment, and here's the question. If a drug is not recommended in renal impairment, that would not go in the dosage administration since no recommendations for dosage modification. Is this correct? Also, does the guidance effectively compel the sponsor to study all groups of renal and hepatic impairment, or can we use the phrase not been evaluated and therefore is not recommended?
So, Ray, I'll take the first question first. That is correct. So, if there's no specific dosage, if there's no difference in the recommended dosage in patients with renal impairment compared to those with normal renal function, that information generally should not appear in the dosage and administration section of labeling. Now, we obviously it may be important to tell people information, provide information about use of the drug in patients with renal impairment. I'm thinking in the context if you have a disease or condition where there is a high frequency of renal impairment, let's say for diabetes, type 1 or type 2, hypertension. Um, in those patients, for example, you may want to communicate how to use the drug in patients with renal impairment because a large percentage of the patient or a significant percentage of the patients have renal impairment. Well, we have a section 8, use in specific populations section. So you can include a separate subsection, essentially patients with renal impairment, and then provide information about the use in patients with renal impairment compared to those with normal normal renal function. So if there's no change in dose in any of the renal impairment subpopulations, that generally would not go in the DNA section. With respect to your second question, so the guidance is really about how to communicate dosage related and administration related information in the DNA section of labeling. It doesn't talk really about the data that is needed to support labeling statements. However, I should say that generally we we do have data on patients with renal and hepatic impairment prior to marketing. However, there's some cases where it's a disease that treats a serious, I should say, or life-threatening disorder or condition, and we don't obtain data on the use of the drug in renal uh, patients with renal impairment or hepatic impairment, and we obtain that subsequently in a post-marketing situation, a post-marketing requirement, for example. In that situation, one can make a statement that the recommended dosage in patients with renal impairment has not been determined, to tell the healthcare practitioner that it has not been determined. So I think that's similar to what was recommended in the statement. Thank you.
Moving on to our next question. Here's the question. In a situation where a product was approved pre 2000s and has information that conflicts with these guidelines, what situation would cause the dosage and administration section to be reevaluated or updated? For example, if the dosing is based on the average dose used in a clinical trial, would that ever be revised?
Good question. So this guidance, the draft DNA section of labeling guidance, is for PLR, physician labeling rule labeling. So if we're talking about a labeling that a product that was approved before 2000 and subsequently was converted to PLR format, then the guidance would be applicable. In that situation, we're not going to recommend changing the recommended dosage. So in this scenario, it looks like dosage was based upon an average dosage used in a clinical trial. If that's the recommended dosage and that was the finding, unless there was a safety issue, uh, there's going to be no recommendation from the FDA to change the recommended dosage. However, with respect to, we potentially would recommend updating the DNA section of labeling. And it's interesting. This morning I was working on a labeling, And this is for a labeling. This is a labeling supplement. It's not an NME. It's not a new biological product. And these are the things that we're going to recommend. Number one, I'm looking at the labeling right now on my other screen. We're going to recommend having important administration instructions prior to the fundamental dosage information. There's also a few statements that doesn't have anything to do with dosage or administration. We're going to recommend that be moved to other parts of the labeling. The titles in subsection in section 2, in the subsections in section 2, basically list the condition. So we're going to, and it's indistinguishable from the titles in section 1. So we're going to say recommended dosage for condition A, and I'm going through the labeling right now. Um, Additionally, there's some statement about what was studied, the study dosage, and as I stated yesterday, the information in section 2 is about the recommended dosage. Information about the study dosage goes in section 14, not in section 2. There's also a statement about usual dosage. As stated in the guidance, we use the term recommended dosage, not usual dosage, because usual dosage may refer to some of what is most commonly used in the community in practice, but that may not be consistent with the recommended dosage. So we're going to use the recommended dosage. So even though we're not changing the recommended dosage, there are things that one can do, one can update, improve communication of the recommended dosage and administration related information and labeling. Thank you.
Thank you for responding to that question. We got a question referring to slide 19, and here's the question. Is there any recommendation or restriction on the type of juice? Can a restriction on grapefruit juice be added here?
Okay, let me look at slide 19. Okay, so the question refers to the use of liquids and or soft solids as mixtures to for administration of certain products. So that we do have a guidance. It's the soft foods and liquids for mixture for drug administration, and that guidance goes into details the different types of liquids that potentially can be used to mix products for situations when you have patients, a certain subgroup that cannot swallow the oral dosage. So, so basically, it talks a little bit, this guidance, about whether the liquid can change over time, the composition or characteristics. Generally recommend selecting liquids or soft foods for that matter, where the composition and the characteristics do not change. So I refer you to that guidance. With respect to grapefruit juice, that is not used, not recommended for mixture because of potential clinically significant drug interactions with several um, CYP-related enzyme interactions. So we generally don't recommend that to use as a mixture, so that wouldn't be applicable there. Thank you.
Thank you for responding to that question. We've got a question regarding the IFU, and here's the question. When there is an IFU for a product, do we have to repeat this information in this section, such as site of injection or time to reach room temperature, etc.?
Hi, Ray. This is a good question about information that should be included in a patient instructions for use versus what should be included in the DNA section of labeling. As stated yesterday, patient instructions for use is written for the patient or caregiver. For example, a parent who's taking care of a child, an infant or a young child, and they're administering the product to an infant, preparing or administering the product for an infant. And patient labeling is written at or below the national reading level, which is at or below a sixth grade reading level. Whereas the prescribing information, the DNA section, is written for the learned healthcare practitioner, the physician, emergency room physician, nurse practitioner, EMT, So the languages used is different. So think about what the healthcare practitioner needs to use for dosage and administration related information. That's in section 2 of the prescribing information. Think about what the patient needs to use. That's in the patient IFU. With respect to, I believe that there was the two items. One was rotation. That seems important potentially for both the healthcare practitioner and the patient. So that would be included in both. With respect to if a product is refrigerated, allowing it to warm prior to use, um, that would be also important, I would think, for from a healthcare practitioner standpoint and a patient. So you would repeat that information in the DNA section and the patient IFU. And there is a redundancy. Um, the patient IFU is based upon the DNA section. It also has additional pictures, diagrams that can be helpful for patients to use. Thank you.
Thank you for responding to that question. Since we're talking about younger children, we do have a question about the pediatric population, and here's the question. For dosage and administration information to be reflected in Section 2 for the pediatric population, do we need a clear indication for pediatrics in the indication Section 1 first?
Great question. So this refers to including pediatric use information and labeling. So we have indications and usage section of labeling guidance that was published in draft a few years ago. And the recommendation there is to include age groups to be very clear to the healthcare practitioner what is the approved population. In fact, the study that I referred to you earlier about the 70 physicians, one of the questions we asked them is if you had a product approved for multiple myeloma, which is a disease condition that occurs mostly in older folks, would you recommend including the age in the indication statement? And the consensus is yes, because one noted that they had a patient that had multiple myeloma who's a pediatric patient. So it's very important to be very clear about pediatric use. And as I stated earlier yesterday, one would look particularly in sections 1, 2, and subsection 8.4. Section 1, indications and usage section. Section 2, dosage and administration section. And subsection 8.4, pediatric use subsection to ensure you have consistent pediatric use information. So in this example, one would include the age group, the pediatric age group, in the indication statement, and we are required to include the pediatric dosage in section 2. In subsection 8.4, we would include the required pediatric regulatory use statement, such as the safety and effectiveness of drug X has been established in pediatric patients, and you can name the age for indication Y. Thank you.
Thank you for responding to that question. We've got a question about a drug with multiple indications and how do we handle the recommended dosage when a drug has multiple indications?
Good question, Ray. So sometimes this is fact specific. Generally, it is fact specific. What we try to do, if the dosage is if the dosage is the same, the DNA section of labeling guidance recommends including the recommended dosage for all those indications, all those diseases and or conditions in one subsection or one place in the DNA section of labeling. One sometimes if they're even if they're different, one can consider using a table. However, if it's extremely complicated, for example, if the dosage is given with different is required to be given with different concomitant drugs, if the administration instructions are different, um, and there's lots of information, then potentially you can include them in different subsections. So it's really a data specific question. Thank you.
Thank you for responding to that question. Got another question referring to slide 27. And here's the question. How is combination therapy distinguished from standard of care in addition to drug X?
So generally, I think you're talking about how to include, when to include information about concomitant therapy in section 2. So generally, this is based upon the clinical studies. So if you have a product for a condition and let's say you have a randomized placebo-controlled trial with the drug versus placebo, and all patients in both groups took um, drug B and drug C, and drug B and drug C are used in that condition, and we don't know the efficacy of the subject drug without that combination. So the combination is really needed, or if it's determined that it's needed for the effectiveness of the drug, we should mention, well, first of all, in the indication statement, we would say drug X in combination with drug B and drug C is indicated for dot dot dot. So we would mention that in section 1, and we would also include that in section 2. We would state that the recommended dosage for drug X in combination with drug Y, drug B, and drug C is, and then we'd provide the recommended dosage for the subject drug. I talked a little bit yesterday about the specificity about what should be included about the dosage in drug B and drug C. Generally, we'd recommend referring out to the drug B and drug C's prescribing information, given the complexity. Potentially, there are dosage modifications due to drug interactions or adverse reactions. There is a different recommended dosage in patients with hepatic impairment compared to those with normal hepatic function. So it's difficult to include all that in the subject drugs labeling. However, sometimes maybe these drugs are being used this new use, and the drug B and drug C's labeling don't include those dosages, then you probably would want to include those dosages in section 2 of the subject drug labeling. As I mentioned yesterday, preparation instructions for the subject drugs labeling concentrates on the preparation of the subject drug. You would refer to drug B and drug C's labeling for those preparation instructions. Thank you.
Well, that's all we have, time we have for questions. We want to thank Dr. Brodsky for his informative talk and also for responding to numerous questions that came in.